Good evening, everyone. Welcome to the Snyder Institute for Chronic Diseases Mini Med School lecture series. We're pleased to host a presentation and discussion this evening on the topic of Creutzfeldt-Jakob disease, mad cow, and chronic wasting disease, deadly and hard to control brain disorders of humans, animals, or both. My name is Hannah Riley, and I'll be the program host for this evening. Before we get started, I'd like to go over a few housekeeping items. There'll be two parts to tonight's program. First, each of our presenters will speak for approximately 20 minutes, and then we will open the floor to audience questions. We'll take a 10-minute coffee break between each speaker, and refreshments will be provided to those of you here in person. You are welcome to ask your questions at any time during the presentations using Slido. To access Slido, you can scan the QR code or type www.slido.com into your browser and manually enter the event code Snyder MMS into the participant field to post your question. Questions submitted to Slido will be viewable to all audience members, and participants can upvote the questions they would most like to see asked. Now on to more specific housekeeping items. For those of you who have joined us online, if you lose your internet connection and drop out, you can return to this session using the same Zoom link you used to join us now. If you experience any technical issues, such as you cannot hear or see the presenters, please email snyder@buxa.com and our team will do their best to support you. For those of you here in person, we ask that you now switch your cell phone to silent mode. Please note this event will be audio recorded, and the recording will be posted on the Snyder Institute website. Now, to formally begin our program, the Snyder Institute at the University of Calgary's Cummings School of Medicine, located in the heart of southern Alberta, would like to acknowledge and pay tribute to the traditional territories of the peoples of Treaty 7, which include the Blackfoot Confederacy, comprised of the Siksika, the Piikani, and the Kainai First Nations, the Tsuut'ina First Nation, and the Stoney Nakoda, including the Chiniki, Bearspaw, and Goodstoney First Nations. The city of Calgary is also home to the Métis Nation of Alberta Districts 5 and 6. And now I'm pleased to introduce our first speaker of the evening, Dr. Sabine Gilch, who is a professor and UCalgary Research Excellence Chair at the University of Calgary's Faculty of Veterinary Medicine. She has more than 25 years of experience in prion research, starting at the Technical University of Munich and later the University of Wyoming. Her research focuses on chronic wasting disease strains, interspecies transmission and zoonotic potential, and PRP genetics to define ways to reduce transmission. She also studies the impact of prion infection on neuronal metabolism with a goal to identify therapeutic targets to treat prion disease. Dr. Gilch has received several awards, including a Canada Research Chair, the UCVM Research Excellence Award, and the CIHR Institute of Aging Mid-Career Investigator Prize. She has published more than 80 peer-reviewed articles, receives funding from national and international agencies, including CIHR, NSERC, and NIH. She is a team lead of a $4.2 million NSERC Alliance Partnership Grant, co-funded by Alberta Environment and Protected Areas, Alberta Conservation Association, Saskatchewan Ministry of Environment, Parks Canada, and NSERC to develop tools to mitigate chronic wasting disease transmission. Her expertise is highly sought after on national and international level. As an advisory board member, co-chair of the University of Minnesota Center of Infectious Disease Research and Policy CWD Contingency Planning Group, or member of the CWD Multi-State Research Consortium. Welcome, Dr. Gilch.
Hey, good evening, everyone. I'm really excited to be here today and to share my passion in research and some of our updated knowledge about prion diseases. So on this first slide, I would just like to summarize that prion diseases are not restricted to one species. So prion diseases can occur in a variety of different mammals, in humans, in cervids, in cattle, also in sheep, and in, as recently discovered, in camels. And prion diseases are also called transmissible spongiform encephalopathy, and I have the explanations for this on the next slide. So even though prion diseases can occur in a number of different species, there are a lot of common features in all of those diseases. So all prion diseases, as the name says, encephalopathies, they affect the brain. So they are neurodegenerative diseases that lead to the death of neurons during the progression of the disease. All prion diseases are inevitably fatal. There is no prophylaxis, and to date there is no treatment or cure for prion diseases. All prion diseases have very long incubation periods, which is the time from infection to the time of the start of clinical disease. And if I say long, this means many years, or in some cases of human prion diseases, even decades, until the disease shows clinical signs. Once the clinical signs starts, this clinical phase is relatively short, and the infected individual will pass away within several months or maybe a year if the duration is longer. In prion disease, I said they are called transmissible spongiform encephalopathies, transmissible because they are infectious diseases. And in some cases, they are transmissible under natural settings, and in some cases, they are only transmissible in experimental settings. And transmission can occur within the same species, so from human to human, for example, or from deer to deer. And in some cases, also between different species, which we will hear about a little bit later when I talk briefly about mad cow disease. So here in this slide, you can see how prion diseases show very characteristic damage in the brain. So this image shows a very thin slice of a brain tissue from a prion-infected individual. And what is very characteristic is this, what we call spongiform neurodegeneration. So during the course of prion diseases, brain cells will die, and then this characteristics holes or spongiform degeneration will appear and will ultimately lead to the death of the host. So prion diseases, as the name says, they are caused by prions, and actually the nature of the infectious agent that causes those diseases was elusive for a very long time. So, for example, scrapie, so what we call the classical form of prion disease, which affects sheep and goats, has been recognized already in the 18th centuries, and shepherds back then even recognized that the disease is transmissible between animals of their flock. However, it was not clear what type of infectious agents caused scrapie, and it took a very long time until the theories that prion diseases are either slow virus diseases or have a connection to bacteria, until this hypothesis could basically be dismissed. Prion diseases are called by this very special agents. And it was established in the early 1990s by Stanley Prusiner that the infectious agents that cause those diseases consist only of prions. And he coined the term prion, which is an acronym for proteinaceous infectious particles. And this is what these prions look like as they are purified from a scrapie-infected brain. So we have this fibrils or rods that only consist of protein. So as opposed to classic infectious agents like viruses or bacteria, which use nucleic acids like DNA or RNA, so genetic information to replicate, prions don't use nucleic acids. So prions only have protein as their information to replicate and to induce disease. And how that works is a little bit like the story of the Dr. Jekyll and Mr. Hyde. So we're talking about the good and the bad prion protein. So every mammal, so everyone here in the room, every animal expresses or produces this host protein. It's called PRPC or cellular protein. It's mostly found in the brain, but also in other tissues. And this is a protein that has a function. It does not cause disease. It's a perfectly normal protein. However, this protein is a shapeshifter. Like Dr. Jekyll, it can convert into the Mr. Hyde form of the protein, which we call PRP scrapie. And if you compare here, PRP scrapie has an entirely different shape. So it turns into this flat molecule, which can interact with others of those molecules that stack on top of each other and then form this tower or this fibrils that we saw on the previous slide in the electron microscopic picture. So basically the good PRPC turns into a molecule that aggregates, that clumps together in the brain. It cannot be digested or degraded by brain cells, so it accumulates in the brain, which eventually leads to the death of neurons and to the patient passing away. So replication now works again without nucleic acid. So the PRP scrapie, the bad form of the prion protein, can, if it comes into contact with PRPC, like the normal form, it induces the refolding or the conversion, like the shape shifting of PRPC into this bad form. So this is an autocatalytic cycle. PRP scrapie interacts, comes into contact with PRPC, it transforms it into the bad form. And this is how it replicates and how they propagate in the brain. So now in the following slides, or for the rest of my presentation, I will focus on animal prion diseases that had the most or still have the most impact on Albertans or Canadians, I would say. And this is on the one hand mad cow disease, on the other hand chronic wasting disease. So mad cow disease or BSE, many of you might remember that it reached epidemic dimensions in the UK in the early 1990s. So BSE is what we call a man-made prion disease, and it was caused by feeding ruminants to ruminants. So animals or cows were fed what's called meat and bone meal, and this meat and bone meal was generated from animal cadavers, sheep, goat, cows. They were minced up, they were pressed into pellets, and then they were fed to the cows. Apparently, there was contamination with prions, maybe from scrapie-infected sheep or cows with prion disease, and this contaminated the food and transmitted prion disease to those cows. So it was kind of a form cows eating cows and sheep, what was called neo-cannibalism, so something that would not occur in nature. What was good about BSE, it was only transmitted through the feed. It was not contagious and not transmissible from cow to cow in the barn. Most cases were found in 4 to 5 year old dairy cattle. And if the animals got to the stage where they developed clinical signs of the disease, they were hyperexcitable. They were very nervous. As you can see, this frequent nose licking is a sign of being nervous. And at the end stage, they had problems with locomotion of ataxia. Basically, they couldn't walk. Their hind limbs became weak and they laid down and they couldn't get up anymore before they finally had to be culled. So this graph shows just the dimensions of the BSE crisis in the UK. So this is the number of cases. This is the year scale. And in 1992, there was the peak of the epidemic. And in the UK, about 180,000 cows were affected by BSE. But in addition to that, almost 4.4 million cows were slaughtered to stop BSE. Unfortunately, the source of transmission or the infection has been identified with being that meat and bone meal. So just by banning feeding ruminants to ruminants, the BSE epidemic could be managed. And you see here, when the food ban was implemented, finally the numbers went down very significantly. In Canada, or North America, there were only a handful of cases, but they were very impactful. And again, many of you might remember that the first BSE-infected cow in Canada was actually diagnosed in Alberta, or it was a cow from Alberta. This was in 2003, and this had huge implications for our beef industry. The US shut down their trade borders, other countries followed, so Alberta or Canadian beef farmers couldn't sell their products anymore, which cost billions every day to the beef industry. So now there is still a surveillance program for CWD, but the latest case of classical BSE in Alberta has been detected in February 2015. So overall, the BSE epidemic seems to be managed because the feed is not available anymore. However, BSE was a problem not only for the beef industry, but also it was transmissible to other species. So there was experimental transmission to cervids, to sheep and goats, and to pigs. And there was natural transmission to exotic ungulates and sioux. They received the same feed, they got BSE. There was transmission to domestic and exotic cats that were fed with BSE contaminated meat. And there was natural transmission to humans, probably also because of ingestion of BSE contaminated meat. There were about 230 cases, and a significant issue with this is that BSE basically proved that certain animal prions can jump this barrier to humans and can infect humans. So prion diseases of animals can be zoonotic. However, again, the transmission now is going down. There are no new cases anymore, because the BSE crisis has been overcome and the disease has been managed. However, now we are facing a new challenge, a new animal prion disease that is very actively spreading across North America. And this disease is chronic wasting disease. So chronic wasting disease is a prion disease of deer, elk, and moose. And as opposed to BSE, it affects both wild and farmed animals. And if you have it in free-ranging populations, this already shows that managing might be very, very challenging. So the clinical signs of CWD it's mostly a wasting syndrome. So if you look at these infected animals, it looks very thin, emaciated. They show unusual behavior, so they lose the fear of humans. And they become also ataxic, so they have problems with moving and walking. Again, as opposed to BSE, CWD is a highly contagious disease. That means it's transmissible from animal to animal. And infected animals can transmit the disease even before they show clinical signs. So, for example, if you look at this deer, it looks perfectly healthy, but for a hunter, it would be impossible to know whether this animal is infected with CWD or not, because it might just be in this incubation period before the disease shows up clinically. And then the animals look like this and they would be identifiable. So why is CWD so contagious? Again, as opposed to BSE, where infectious prions were mostly confined to the brain and the central nervous system, in CWD, we find infectious prions basically throughout the body of an infected animal. So we have infectious prions in skeletal muscle, in antler velvet, and infected animals shed infectious prions in feces, in urine, and saliva. And this all contributes on the one hand to direct animal-to-animal transmission, but also it results in the contamination of the environment with infectious prions. And prions are very sturdy. They can last as in their infectious form, bound to soil or attached to plants for many, many years, at least 5 to 10 years, and still remain infectious if a deer comes and takes up the right amount of soil. So there is both direct and indirect transmission of the disease. And this made CWD very successful, not a nice success though. And this map shows the current distribution of CWD. It's been found in 36 US states, in 5 Canadian provinces, with British Columbia being the latest that has identified cases of CWD only last year. And if you look at this dark gray areas, this is the distribution of CWD prior to the year 2000. And since then, it has spread all over. So the gray areas is CWD in wild, free-ranging animals. And the yellow and red dots is depicting CWD-infected farms that either have active, that remained active or that depopulated their animals due to CWD. So it's spreading and it's not going away. So in Alberta, in wild cervids, the prevalence across all samples that are submitted to the provincial surveillance program is about 19%. That means if like 100 animal heads of deer are submitted, about 19 will come back positive for CWD. However, this is not the same across the landscape, and across the species. So CWD is mostly found in mule deer in Alberta, where it reaches a 30% prevalence or positivity rate. But there are hotspots in Alberta in the southeastern corner at the border to Saskatchewan, where the CWD positivity rate in male mule deer exceeds 50%. So every other mule deer buck in those areas is infected with CWD. And that's really significant because we get into numbers where CWD affects the cervid population. So if the percentage of infected animals reaches a certain number, like 20-25%, then it will affect the population. So the number of deer in that population will go down. And we also have CWD on deer farms, and this is just a list from the CFRA. It's a reportable disease. And since 2019, so over the last 5 years, 47 new farms were identified that have CWD in their animals. And it's important to note that there is transmission. So farmed animals and wild cervids, they interact. So there is transmission from farm to wild and vice versa. So it's not isolated systems. So with this shedding and the passion for hunting that many people have, of course, the big question is that CWD as BSE was, is it transmissible to humans as well? And this is some research that we are very actively following on here at the University of Calgary. So there are different ways to approach that. So on the one hand, there are epidemiological studies which look into the numbers of human prion disease cases in areas where CWD has been around for a very long time, and whether this case numbers increase over time, which could be an indication that CWD is a contributor of that. However, these studies don't show any evidence that CWD has been transmitted to humans. There are systems that test in a test tube whether the CWD prions can induce the conversion of the human form of the normal prion protein of the human PRPC into CWD. And there are studies that say, yes, it's possible. Then there are, of course, animal models that we can use that are close to humans in some way to test whether CWD is transmissible to those animal models. And non-human primates are probably on top of the list in terms of they are similar to humans. They have been used to show that BSE or to contribute to the understanding that BSE causes prion disease in humans. And there are two types of non-human primates that are commonly used. So on the one hand, spider monkeys, and these were perfectly susceptible to CWD when they were either fed with CWD-infected material or the material was injected directly into their brains. Then there is another model which is considered closer to humans, and these are cynomolgus macaque monkeys. And there are two studies that show conflicting evidence. So one that has been published from the US says, no, there is no evidence for zoonotic transmission. And another one that's a UCalgary-led study with an international consortium of researchers says, yes, there is transmission, but it's a very atypical disease that is produced by CWD in those monkeys. And then what we've also used are transgenic mouse models that were engineered in a way that they express the human PRPC. And I will call them humanized mice and the following, and they can also be used to model the potential for transmission of CWD to humans. And we've done that in the lab. So we used 2 CWD isolates from deer. So we took their brains, minced it up into homogenate, and injected those homogenates into the brains of the humanized mice. And we waited for, almost 3 years in total. And we monitored the animals, whether they would develop clinical signs of prion disease. And about 75% of those animals indeed showed clinical signs of prion disease. However, they were very different compared to what these animals would develop when they were infected with real human prions. And the clinical disease started very late, about 1 to 1.5 years after infection. And then the progression of the disease was very, very slow, which was also a very unusual feature. Once the animals were euthanized, we took their brains and we looked for the classical markers of prion infection, which is basically PRP scrapie. And when we did this, again, we found very little evidence. And what we found was, again, very different or atypical to normal as compared to normal prion disease. And this is shown here. So this is what PRP scrapie looks like in a normal prion disease or in scrapie. And this is what we found in the brains of those humanized mice. So again, very atypical patterns. And we had to use amplification assays to detect prion infectious prions or prions in the brains of those humanized mice. And with these highly sensitive amplification assays, most of the mice were positive. And finally, what was really striking, we tested the feces of the humanized mice that were infected with those CWD prions. We used again this amplification assay and they were positive. So the question was, is this real? So is this a false positive result, or do we really have infectious prions in the feces of those humanized mice? So we transmitted homogenates again to a rodent model. These rodent models developed disease, so we indeed were able to show that humanized mice infected with CWD shed infectious prions in their feces. This has not been shown before for any other human prion disease that was propagated in this model. So what does this mean now? So for sure, this is not a proof that CWD has been transmitted to humans or will ever be transmitted to humans. However, it shows that the barrier might not be absolute. So it might be possible. And if CWD transmits to humans, it might show up or present as a very, very different disease as one would expect. And it might be really hard to diagnose the disease with the standard tests that are currently used to confirm prion infection. And for the infectious prions in the feces of CWD humanized mice, of course, the question is, if CWD is transmitted to humans, would they shed prions in feces? Would CWD become a contagious disease among humans? But these are questions we cannot answer. We can only say, don't eat, if you hunt, don't eat CWD positive meat. That's the current recommendation for Health Canada, because we cannot fully exclude that there is no risk to humans. So from this, you might have seen CWD affects animal health, environmental health, human health. So it's a very classic One Health problem. And what we urgently need are mitigation strategies or management tools for CWD that are widely accepted in the communities. So I have the privilege to lead a large team grant bringing together researchers from the University of Calgary, Alberta, Saskatchewan, with partners from provincial governments, provincial wildlife organizations, federal agencies, and Indigenous community partners to develop mitigation tools or a toolbox for CWD, with oral vaccines being very much at the center of this toolbox. But we're also exploring a combination of different approaches, like looking into genetic resistance of deer, and environmental decontamination to finally end up with a reduction of transmission of CWD, protect cervix, and reduce the shedding and the ongoing spread. And in my last slide, I would just like to leave you with kind of a silver lighting, or the question, is there light at the end of the tunnel? And the data I'm now showing are from the Schaetzl lab. They are heavily working on developing a CWD vaccine, which is not an easy task because the immune system overall is blind to prions because it's a self-protein. So they have to use some tricks to get an immune response against prions. So they found those tricks. They use currently cervidized mouse models, again, genetically engineered to be susceptible to CWD. And they found that with vaccination, they can extend the survival of the vaccinated animals. Moreover, now they have really recent unpublished results that go a step further. If we have an extension of survival time, is this good or bad? Because if the animals shed the same amounts of prions in feces, we would just increase the environmental load with prions. So they looked into feces, how vaccination affected the amount of prions in the feces of those animals. And indeed, vaccination reduces CWD prion shedding, so less animals were shedding, and those that were shedding had less prions in their feces. And this is now really a very important finding. So finally, ongoing work. So of course, the next step would be to transfer this mouse work into the real host, into elk and deer. And we have several vaccine trials going on in elk, in white-tailed deer. And the final goal is to develop an oral vaccine that can be deployed in wildlife. And there are also promising developments on that front. And finally, I would just like to introduce our dedicated troop of young scientists at the Calgary Prion Research Unit who work tireless and do the actual work I was just presenting. And thank you for your attention.
Thanks, Dr. Gilch, for the wonderful presentation. We'll now take a short break for coffee and we'll resume in 10 minutes. Welcome back, everyone. I'd now like to introduce our next speaker, Dr. Hermann Schaetzl, who is a professor of prion biology and immunology and associate dean of research at the Faculty of Veterinary Medicine. His work over 30 years focuses on the biology of prion diseases, fatal and infectious neurodegenerative disorders in man and animals. Trained by Stanley Prusiner, winner of the 1997 Nobel Prize in Physiology and Medicine, he established his own laboratory at the University of Munich and Gene Center Munich in 1995. From 2002 to 2010, he was head of the clinical virology unit at the Technical University of Munich. In 2010, he was appointed Wyoming Endowed Excellence Chair in Prion Biology, and in 2013, he joined the University of Calgary to set up the Calgary Prion Research Unit. He has supervised more than 140 trainees and acquired approximately $30 million of external funding for his lab. His current research focuses on the zoonotic potential of chronic wasting disease, an expanding prion disease of cervids, and on developing vaccines against CWD. Welcome, Dr. Schaetzl.
Thank you very much. So it's a privilege to be here and to talk to you, and thanks for being here. So the topic now actually is more on the human prion disease side. So as Dr. Gilch already mentioned, prions are coming in 3 flavors: acquired by infection, sporadic from inside, and also genetic from inside. And in human prion diseases, we have all 3 flavors. So we'll talk a little bit about Kuru and iatrogenic CJD, and I will tell you what this stands for, and variant CJD, which comes from BSE. The majority of human patients actually are sporadic CJD patients, where we think we have a spontaneous change in the prion protein conformation. And in humans, we also have genetic forms of prion diseases. About 10% compared to the 90% CJD comes within families where we have usually a change of one amino acid into another one, which makes it from normal into pathologic. So pictures again, sporadic CJD, and CJD actually stands for Creutzfeldt-Jakob disease. It was described in 1921 by Creutzfeldt and Jakob, two German neurologists. Kuru, I will talk a little bit, was described in 1957 by Carlton Gajdušek, who received the Nobel Prize in 1976. And variant CJD is a more modern human prion disease, which was first described in 1996. So human prion diseases, as I said, sporadic, familial, and acquired. Sporadic CJD shows up worldwide, pretty constant, with a rate, with an incidence of 1 in a million. So this means in Canada, we have roughly 40 patients every year newly diagnosed, and they're going to die because this is a fatal disease. When you look now how many at the world population, which is, I guess, 8 billion or so, this means we have about 8,000 people every year dying on sporadic CJD worldwide. And over 10 years, these are 80,000 or so, right? So that's quite normal, 1 in a million. The familial form are about 10-fold less frequent, and the acquired forms are rare, and we will talk about them. Kuru was caused by a ritualistic form of cannibalism. Variant CJD was caused by BSE. Also, we do not really understand how this was actually happening. And this form actually is variant CJD within humans. So all these forms have as a common feature a very long incubation time, a very short clinical phase. They are always deadly. We don't have a preclinical disease with one exception. The familial form we can pretty easily diagnose, and there is no therapy. So now I will zoom in a little bit on the manifestation, the clinical phase, and the incidents basically attached. So sporadic CJD is characterized by a progressive dementia. Dementia basically means a chronic decline in thinking, memory, reasoning, etc., because it affects the cortex. So familial forms, CJD, these are two other names, Gerstmann-Streissler-Schenker, and fatal familial insomnia. Again, progressive, but here it's more the cerebellum, it's ataxia, which in the cerebellum we need for coordination, muscle movements, speech, and such things. The iatrogenic form, human growth hormone and duramata, I will show you later what this is. They are acquired, and here it's again the cerebellum, progressive ataxia. Between 200 and 300 cases are known. Kuru was also ataxia, the cerebellum, and the estimates are around 4,000 to 5,000 cases in the Kuru region. And variant CJD, the most recent form, actually starts with psychiatric syndrome. Very different, and we have at the moment 230 cases. So as you have heard, there's a really long incubation time, a very short clinical phase. So sporadic CJD, usually the patients show up around 60, 65, and the clinical duration is around 3 months. So this is basically Alzheimer's disease, which is 10 years and more, reduced to a few months. And it's progressive. It's always getting worse. So basically the patient develops symptoms, sees his doctor, is transferred into a hospital, goes to neurology, and a few weeks later the patient is going to die. So really, really dramatic. So this is normal 2 to 4 months. The familial forms come a little bit earlier, around 50, and the clinical phase is actually longer, 1 to 2 years. But you have to remember the mutation in the prion protein is from birth. So it takes basically 50 years until such patients develop symptoms, and again, they're going to die. So the acquired forms, iatrogenic CJD. Iatrogenic means caused by the physician. That's an old ancient Greek work, iatros, the physician. So caused by the physician. So we know the youngest patients, 4 years, more than 40 years incubation. Kuru was pretty well studied. The youngest Kuru patient was 4 years. The oldest one was over 60 and most likely was exposed as a very young child. So it takes a very long time. Variant CJD, more than 10 years. It came 10 years after the BSE peak and 1 to 3 years the clinical sign. So sporadic CJD, as I said, rapidly progressive dementia. And these are the neurological signs, myoclonus, cortical blindness, so they don't see anymore. Then all kinds of neurological signs, extrapyramidal, pyramidal, etc. This actually stands for not moving and not speaking, not hearing. So it's almost like a lock-in syndrome and so. So I show you this because there's a little bit of a risk factor, but the risk factor is in the prion protein gene, in the gene encoding the prion protein. So Caucasian population has 2 amino acids, either methionine in 40%, methionine, valine in 10%, and 50% have a methionine and a valine. This is the normal case, but in sporadic CJD, the blue, and this is variant CJD, we see mostly NVV patients. So people, and this is 50% usually, who have a methionine valine, are a little bit protected, but it's only a relative protection. They usually show up later when they are older, but it's some kind of protection. So Gershman, Streisler, Scheinker again was described in the 30s. These are the people describing it. GSS is a genetic form. It comes within families, and depending where the families actually went, you find it worldwide. But all kind of families reported. So it's an autosomal dominant disease. And previously we thought whenever you have the mutation, you will develop the disease. That's actually not true. So people looked a little bit more carefully, and depending on which mutation you have a higher risk or a lower risk, and there are mutations you might not develop a prion disease. So it depends a little bit. So as I said, a longer clinical course, it's a little bit different from sporadic CJD. And here we can pretty easily diagnose the disease because we look for the mutation. You even could do prenatal diagnosis, but I'm not aware of any country which really recommends it to do prenatal disease for this, prenatal diagnosis for this disease. I just mentioned this genetic disease, fatal familial insomnia, very rare, was described in the 80s, but as the name says, insomnia is the leading syndromes. These patients cannot sleep, so they have a very profound disturbance in their sleeping behavior mostly. So now I move on to iatrogenic, which means by medical examination or treatment. And such patients usually have a risk factor, which is the history. For example, receiving human growth hormone, tissue grafting, or having a neurosurgery, for example. So, and these are the risk factors. Human growth hormone, before it was by recombinant technology, was coming from cadavers. So it was cadaveric derived human growth hormone. It comes from the pituitary gland and so. And usually a batch of the hormone was made by a few thousand patients. And it occurred a few times that the Creutzfeldt-Jakob disease patient was among the people and contaminated the entire batch. And these were usually children receiving the growth hormones or gonadotropins. So there are more, close to 200 cases. So whatever literature you look at. Another risk factor is human dura mater, which is the skin of the brain, which again was coming from cadavers, mostly in pathologies and so. And this is used for surgery of the big blood vessels in the brain and outside the brain. And more than about 150 patients were actually contaminated by receiving such a dura mater transplant. Then there are a few patients with EEG electrodes, actually 6, and a few patients with cornea transplants. And then more modern blood. Blood is a risk factor for variant CJD. So now Kuru, actually the name in the language of the four was found in Papua New Guinea, in the Pori region where the Okapa people actually were living. And it was the leading death in this area. And it was also called the laughing death. So they were very happy, the patients and so, and mostly children and women. And this boy actually was completely healthy 5 months before the picture was taken. So within 5 months, the boy couldn't stand anymore, couldn't move anymore, and you see the wasting. The symptoms which Dr. Gilch described for chronic wasting disease are pretty much the same as you see it in Kuru, wasting and others and so. So it was caused by ritualistic cannibalism. It was a form of honoring actually their people and so, and mostly children and women participated in such feast. It was never documented. There is no movie showing this, but it's pretty sure. This was actually, when it came to Australia, they stopped this practice, and then actually the transmission stopped, and the latest patient actually showed up in 2006. But this patient was most likely infected in the 50s. It took so long until the disease showed up. So now I switch to zoonotic prion diseases, and Dr. Gilch already described. And this is actually the story of BSE in humans causing variant CJD. And I guess she already said it. The terminus here actually is neocannibalism. And then it happens. So there's a list of human prion diseases. Of course, Kuru, whenever you eat humans, don't eat a Creutzfeldt-Jakob patient. Very bad idea. That's probably how this started here. Iatrogenic, not on purpose, right? Variant CJD has to do with BSE, and then it was transmitted within humans. So that's the situation. Only 233 cases are known, but huge impact in the 90s and the last years. Many of you were not allowed to donate blood, for example, when they were in UK at that time. A lot of money because they changed how they did the surgeries, and so a lot of impact on the health systems. 233 cases in 12 countries, mostly in UK, quite a bit in France, 4 in USA, 2 in Canada, but these are imported cases that most likely were in UK at this time and so. And they were all young. The clinical course was different. They all showed up in with psychiatric symptoms. And the pathology in the brain was very different from sporadic CJD. And there was no real exponential increase. So the age around 25, 30. This is sporadic CJD around 60. So very different. And actually, the worst case scenario happened. Variant CJD was people actually did blood donations before they were sick. About 10% of later variant CJD patients were blood donors. And at least 4 persons were infected by blood donations and 1 by factor VIII blood products, actually. So that's the risk. So, Dr. Gilch started about the zoonotic potential of chronic wasting disease. I will not go into this, but we finally will come up with our manuscript. It took us a long time. So this study goes back more than 15 years. But the conclusion will be that we see some zoonotic potential. Whether this ever translate into human cases, we don't know. But this started with cynomolgus macaques, and then we did transmissions into different rodent models, and we see some transmission. But this is all what I say. It's not published yet. So the final thought, actually, there's a prion-like behavior. You might have heard that in Alzheimer's disease, in Parkinson's disease, in ALS, and also in Huntington's disease, which are much more prevalent, people talk about a prion-like behavior. There are key proteins which also go from normal into abnormal, into pathologic, and they behave like prions a little bit. The real difference is they only go from cell to cell, from tissue to tissue sometimes, but not from individuum to individuum. So no Alzheimer patient really is infectious to another one. So that's the difference. But otherwise, there are many similarities, and actually Stanley Prusiner calls them all prion diseases, right? So this is actually a slide I made from the 2024 Prion conference, which was in China, and he says there are prion diseases, Alzheimer's is a prion disease, Parkinson's disease is a prion disease, the protein here is alpha-synuclein, here the proteins are tau and A beta, and he calls them prions. The field does not agree, so we still prefer the terminology prion-like, not prion, right? But this is an ongoing discussion. Actually, I have a few more slides on diagnostic and therapy. So diagnosing prion diseases is not easy, human prion diseases. And in the old times, we really looked for p-episcrapy in the brain. There's no antibody reaction which you can measure in the blood, so it was complicated. But now more and more people use MRT and so, and also a little bit CCT and PET scans and so. This helps a lot now in diagnosing prion diseases. CSF is very helpful diagnostically, but you need a spinal tap, so that's invasive. And then you can look for marker proteins, which are kind of biomarkers, also will be important for therapy and so. But they are basically proteins which show up when many neurons die. So they're not specific. It's basically neuronal cell death marker. But what really is fancy, we have a kind of a protein PCR, ultra-sensitive technologies. There are 2 different ones which amplify prions to really a level that they are detectable and so on. And Dr. Gilch also mentioned this. So this has the power to become a blood test and is also now used for tissues, sometimes tissue outside of the brain. So this has a lot of potential, not just for prion diseases, also for other diseases. So now the last one is therapy. So in principle, we have a normal protein misfolds into the pathological ones, and then you have this cascade, and it's an autocatalytic process. So you could interfere here, PRPC, which is the substrate. You could take away the product, and so which is the template for the reaction, and you could define something which interferes here in the process of prion conversion. So in theory, simple, right? You have clear targets and so, but the reality is it doesn't work so easy. So there was a lot of work in the last 40 years and so, and our groups also invested a lot into this. So a lot works in vitro and in cultured cells, but most of the drugs are toxic. They have side effects. The dose which you need in the brain either is not possible or you have side effects again. They have to penetrate the blood-brain barrier. It's a disease of the central nervous system. Sometimes people use pump system to bring it to the central nervous system. So at the end, there are only a few human trials, a few drugs were used, and none of them really was effective in the small human trials. So we don't have a candidate really for therapy. But things changed a little bit with this person. So this is Sonia Valab, and she's a patient researcher. So her mother died in 2010. The mother was 52, and it was a mysterious brain disease. And a year later, Sonia was diagnosed with a familial form of CJD, which means she is going to die, depending on the mutation. But she changed her life and started retraining as a Prion researcher. So she did something completely different, but she actually started this, and together with her husband, who did the same, this is a real success story, right? So she's now a group leader in MIT Harvard in the Broad Institute, and her husband is an assistant professor there. They're well known, and they really look into familial diseases, into therapy, into diagnosis, and they push our field because they are so pushy, right? They really try things which we said, well, doesn't work. They really go into it and push it. And this was one of the first paper in 2019. They used a very simple thing, antisense oligonucleotides. Very old strategy. They used it in mouse models, and what they found actually is when they reduce PRPC in the system, using antisense RNA or modern gene editing, they get extension of survival up to 50%, which is fantastic in our field. And they improved this now with better substances, better approaches. And the first clinical trials are going to start soon. And there were 2 more publications last year and this year about gene editing strategies. And this is really light at the end of the tunnel, right? And other neurodegenerative fields are also using now similar approaches, taking out the protein which is going to misfold. So this is a real success story. So I stop here with this slide, and this starts again with BSE. The problem here, we will always deal with sporadic diseases. For example, sporadic BSE, sporadic Creutzfeldt-Jakob disease, and so on. And then you can have transmission within a species, and usually you need human help. And then you have an epidemic form. Sporadic goes into epidemic. When you do a very bad job or have very bad luck, the epidemic form in one species can transmit into another species. And this happened from BSE into variant CJD in humans. And even more bad luck, variant CJD again found an interspecies transmission, iatrogenic via blood donations. And now you have an iatrogenic form of this disease. So what I want to say, we never will get rid of prion diseases because they come spontaneous, sporadic. What we have to do, be careful, put a lot of precautions into not coming into such scenarios, right? We cannot get rid of this one, but we can get rid of this one. So I stop here, and I also thank our people in the lab. They are fantastic. That's the same picture, I guess you should. And our colleagues and funders, right? And usually I stop with this one. So.
All right. Thank you, Dr. Schaetzl and Dr. Gilch, for the fascinating presentations. We will now open the floor for a Q&A period, and we'll do the best to alternate between questions from Slido and our live audience. As a reminder, questions can be submitted virtually through Slido by scanning the QR code or by opening slido.com and entering the event code Snyder MMS into the participant field. For our live audience, please raise your hand if you have a question, and we will pass you a microphone. We'll do as many, we will try to answer as many questions as possible in the time remaining. So while we get set up here, I'll take the first question from Slido, and it is CWD transmissible to predators such as cougars?
Yeah, maybe I'll take that one. That's a great question. There is no evidence at the moment that CWD has been transmitted or transmissible to any predators. There were experimental studies that looked into to model potential transmission specifically to cougars, where they again took CWD prions, injected them into the brains of domestic cats, and they developed prion disease, but of course it's a very artificial way of introducing prions. But in nature, there is no evidence that any predator got CWD to date.
Thank you. We can now take a question from our audience. So we'll go over there.
Thank you for the presentation. That was really enjoyable. I noticed you didn't mention species at risk, including antelope or bison. And then, of course, in Alberta, you're starting to see quite a few llamas and alpacas. Are they at risk as well?
Yeah, again, great question. And this is certainly an area that needs more investigation. So looking into species that live in the same areas as CWD-infected deer. There are indications from lab experiments, again, using rodents that were engineered to express beaver PRP or the antelope PRP. And again, if these mice are injected directly into the brain with CWD prions, they become sick. However, the big question is always, is this possible under natural exposure, like a beaver eating CWD-infected parts or an antelope taking up CWD prions from the soil? Again, there is no documented evidence, but I think there is also not a good surveillance or testing system really looking into samples of other wild animals other than deer. And part is, again, it's always about money at the end of the day. Alberta has a great CWD surveillance system for deer and elk, and they really work hard to keep it up. But I guess test a lot of other like non-cervids where there is no documented risk for getting the disease is probably just too costly as well.
Thank you. Our next question is, can hunters test their harvested meat for CWD? Share the one mic. Can hunters test their harvested meat for CWD before consumption?
Yeah, so again, there is, Alberta has a very good surveillance system through the government, and there are specific hunting areas where you have to submit your deer head for sampling and testing for CWD, but there is always the option to voluntarily submit the head of your hunted deer to get it tested by the government. In other provinces, there are also labs who can conduct the testing. For example, in Saskatchewan, it's completely voluntary, so every hunter has the opportunity to submit the head and get the test results.
If someone in the audience has a question, I'll let Anu decide who to give the mic to.
Thank you so much for the presentation and all of the information you brought. I just would like to ask, as a high school student, why don't the antibodies react to the prions there? Does it have something to do with the prions being proteins? I just saw that mentioned in a slide.
Yeah, that's a very good question. So basically the principle prions are using whenever they go into a new host, for example, when you eat BSE, and then the prions which are made in your body will be basically human prions, right? Because it's the human PRPC which is used as the substrate. So the prions which are generated in the bodies actually are self. And that's the problem. The immune system basically considers PRPC as a self-protein because it's a highly expressed protein in the brain and other sites in the body. And the immune system also considers PRP-scrapie as self-protein because it's a protein. And apparently our immune system cannot discriminate between the good and the bad form. Also, they are so different in their biophysical properties, but the immune system actually a large part of the immune system reacts on the protein level for amino acids, peptides, and so on. And that's why the immune system apparently cannot well discriminate between PRPC and PRP-scrapie. There might be exceptions, but overall, I would say it's a problem.
Thank you. The next question is, after experimental transmission of BSE from cattle to deer, were the characteristics the same or did they look more like CWD?
So fortunately, they look different. So the characteristics in the brain, the distribution of prions in the brain is different. And also the PRP-scrapie, the biochemical signature is different. So for most cases, or what has been predicted, is that it might be possible to distinguish CWD infection in cows from BSE. But the problem again is the animals were mostly susceptible if the CWD prions were introduced directly into the brain, and not so much if cows were fed with CWD prions. So this is always the balance. We do a lot by injecting prions from one species directly in the brain of a model for a different species, just to show in principle whether it's possible for the CWD prions to convert the PRPC of another species. But this does not yet tell us whether a real natural transmission might occur.
Thank you. If someone in the audience has a question, I think someone already has a mic.
What's the normal function of PRPC? And you mentioned that sometimes the error occurs in the cerebellum, sometimes in the cerebrum, sometimes in other tissues. So what is a PRPC that's found in different sites in the body? Does it have different functions?
That's an interesting question. Actually, all questions are interesting here, I must say. And that's a very tough one. So our field, since many years, we try really to come up with what's the real function of the prion protein. And so far, we cannot find a consensus what it is, right? There are many functions postulated, and the most likely one is somehow neuroprotective. The problem really is PRPC is in the outer membrane of cells, and how it transduces into the inner side of the cells. It can't actually. It needs interacting proteins. We have some ideas, but we don't really know what's going on. So, and when we make a knockout mouse, which sometimes tells you what's the function because you destroyed the protein, you knocked it out. The knockout animals where we remove the prion protein look pretty normal, right? So apparently it's a non-essential function when you knock it out or it's compensated. So, but we don't know. Your question now, whether there are different functions, whether it's the cerebellum or the cortex, we don't know, but we talk about selective vulnerability in the brain. And depending what kind of prions you have, we see differences. So sometimes there's more destruction here and there, but we do not yet understand the molecular mechanism. So this is not a good answer, but do you have a better one? Yeah. But the good thing is PRPC, you can take it out of the system. It's a good target for therapy and a good target for vaccines, right? Because interfering with the function of PRPC apparently does not produce lots of side effects. So that's a good thing, actually.
Thank you. The next question is, if prions from cows were confined in the brain, how did humans get infected from their meat?
Again, a very good question. So brain means everything which is part of the central nervous system. This includes the spinal cord, the eyes, and such things. And before 1989, I guess, everything from a cow was going into human food, right? So only then actually they stopped actually putting the potential infectivity in a cow. The central nervous system, as I said, spinal cord, brain, and which is part of the central nerve, into human food supply. So this basically means BSE was mostly a problem before 89. Or was it 88, 89? So, and later when they removed the potential infectivity, it was much better. And you could see it actually. So probably most people who developed variant CJD were infected before. But before this change in behavior in UK and elsewhere, as everything was going into the human food chain.
Thank you very much for the talk. I feel I came in here not knowing what a prion was, and I have a slight idea now. But just in the last little bit of your talk, you brought in the subject of Parkinson's and ALS. Are they also caused by misshapen proteins in a similar way, or are they completely different and just happen to have a relationship?
It was in my presentation, yeah. So I would say short answer is yes. So it's the same principle. You have a protein, which is supposed to be a normal protein. The protein changes the conformation. It starts to aggregate to form higher forms, and apparently this causes toxicity in the system. It's the same principle, right? A protein changes the form, the shape, and goes into another one. So this is a prototypic misfolding diseases, right? And it's always a protein which misfolds into another protein. So it's the same principle, and this is why we also say prion-like, because such conformers can start in one cell and they can transfer into another cell and start the same process. It's not so well understood, but you can observe this process. The difference is there is no infectious process from one individuum to the other one. Also, there were a few reports which say otherwise actually for Alzheimer's disease, but let's say in general, no. Exactly.
Thank you. The next question is, do clinical symptoms of CWD begin showing faster than prion diseases that are less contagious?
No, I would not say that they would show faster. It's always the same thing. You have many years of clinically silent incubation period, and then clinical signs start to show and they progress rapidly. I mean, in wild deer, the thing is that most CWD infected animals don't reach that terminal clinical stage because in the disease progression, they slow down, they become less aware of their environment, and so they are preferred roadkills or preferred prey for cougar or other predators. So it's quite rare that really a wild deer goes up to the stage where they have clinical CWD and die for it. But overall, I would not say that they show faster. Show faster than prion diseases that are less contagious.
Whoever has the mic in the audience can go ahead.
That would be me. Thank you, both of you. Very interesting. I'm curious about the blood supply. So when people go to give blood, we're asked to self-select. So was I in the UK? I can't remember the questionnaire how long ago. But the disease takes a long time to present itself. So I'm wondering, is that a good method? And is there do they then test the blood? Can they test for Creutzfeldt-Jakob? Or what can you tell me about the blood supply here in Canada about these diseases? Thanks.
Okay, so I'll give it a try. So the question about the UK, that was basically being in the UK during the BSE crisis, that was the main risk factor for getting infected with BSE prions or to develop VCJD, and that's why this was the critical question. And at the end of the day, there is not much more than you can do than saying, okay, you were there, so unfortunately you're not allowed to donate blood. Actually, this has been lifted in Canada for, I think, last year. So now, even if you were in the UK during the BSE crisis, you're now allowed to donate blood again. And that probably has to do that now people feel there is enough time has passed. If you would have developed VCJD, it would have happened. As for the blood test, there is, and the blood supply, there is no test at the moment that's being used, but those in vitro amplification methods, they are getting more and more sensitive and more precise in their ability to detect very low levels of prions. And like one application definitely is to test blood donations and to make sure that the blood supply is safe, but they have not yet been implemented as routine tests for blood supply.
I mean, the specific problem with testing blood is you do not want a lot of false positives, right? So that's a huge problem when you do such tests. So you want highly specific, sensitive, but highly specific tests. And usually they pool it when they look for viruses and so. And that's the problem. When you have a test which shows 2% positives, you're in big trouble, actually.
Thank you. I realize we still have a few questions, but unfortunately we're out of time for tonight. But before we go, I just want to thank our presenters, Dr. Gilch and Dr. Schaetzl, the Snyder Institute for Chronic Diseases, Buxa, the Cummings School of Medicine, and each of your audience members for attending tonight's mini-med lecture. We hope that you enjoyed it and invite you to join us on April 7th for our next lecture on the health effects of wildfire smoke. Thank you, everyone, and have a great evening.